Hello, my name is Dr. Kirsty and I'm a teaching fellow at Imperial College London. I'm going to be teaching on the new BSc in Medical Biosciences, specifically the first year module, Molecular and Cellular Biology, and I'm also going to be involved in the lab pods. So I thought I'd use this video as an opportunity to introduce myself and to tell you a bit about the research that I'm involved with. I finished my PhD at the University of Sussex about 6 years ago and decided to move to London and join Imperial College as a postdoctoral researcher. I've always been really interested in the field of epigenetics. This is where we study the mechanisms that control gene expression in a cell without actually affecting the DNA code itself. So in the first few weeks of the molecular and cellular biology module, we're going to be covering this transfer of information from DNA to RNA to protein. And I'm not going to be able to resist telling you a bit about how epigenetics fits into all of that. However, you'll learn much more about epigenetics later on in your course. So I think epigenetics is really interesting because it's controlling which genes are switched on and switched off. And when you consider that almost every single cell in your body has exactly the same DNA, but they don't all look the same, they don't all act the same. So it becomes very important which genes are switched on and which genes are switched off for each of those cells to do their jobs properly. In the same way, you could compare the epigenetics in a normal cell to a cancerous cell and find out what changes have happened that have caused that cell to become cancerous. So for example, a gene might be switched on that is continuing to tell the cell to grow when it shouldn't be. Or it might be that a gene has been switched off that actually should be stopping that growth in the first place. So these changes in these cancerous genes that are known as oncogenes or tumour suppressors can be very interesting epigenetically. So you'll learn a lot more about tumour suppressors and oncogenes in your second year module, cancer biology. So that's what my research has always been based on, is these epigenetic changes between normal cells and cancerous cells. But I've also been looking into the changes that occur epigenetically between cancer cells that are sensitive to chemotherapy treatment and those that aren't, those that have become resistant, looking at what changes in gene expression have caused a resistance to these drugs. It's especially interesting now that there are drugs available that are epigenetic modifiers, and so they could potentially reverse the changes that have happened epigenetically and allow that cancer to become resensitized to their treatment. There's also been a massive advent of new cancer treatments that have come out recently. Including immunotherapy. It's been a big breakthrough recently, which kind of harnesses your body's own immune system to treat the cancer. But some people really don't respond to it. I've been looking into potential reasons for why they might not be receptive to this kind of therapy and how we might be able to modify this by combining that treatment with other drugs. So if you want to find out a bit more, I've written a blog post which goes into a little bit more detail about this. I'm going to wrap up there. Thank you very much for watching my video. I hope it was interesting to you, and I look forward to potentially meeting you in the near future. Thank you.